Greetings and welcome to another lecture in Introductory Psychology. This one involves how brain cells send signals one to the other, and it's called the nerve impulse or the neural impulse. And I gotta tell you, this is kind of complicated. So the odds are you're going to be watching this one perhaps more than once. You may want to try to make heads or tail of it in your book. I'm going to try to present it both verbally and using a picture, a diagram. You may want to go back and relook at one once you saw the other. But at any rate, first I'm going to go over it in words and describe what happens. Now, this neural impulse is how signals are sent from one neuron to another, whether it's in your brain, spinal cord, peripheral, central nervous system, whatever. Now, when neurons are not sending a signal, they are said to be resting. They have a resting potential. They have the potential to send a signal, but they are not sending a signal because they're resting. Now, when a neuron is resting, it is more negative on the inside than it is on the outside. And that is because of our players. There are three chemical ions that play a big role here. It's why the signal is called an electrochemical signal. It involves both chemical ions as well as electrical charges. And the three that we're talking about are sodium, potassium, and chloride. Now, sodium tends to be abbreviated Na. Used to be called natrium, and when they changed the name to sodium, they didn't change the abbreviation. Potassium is abbreviated as K, a capital K, because it used to be kalium, and again, when they changed it, they didn't change. And chloride is Cl. By the way, this little discussion may also show you why salt is very important in our diets, because the chemical formulation for salt is sodium chloride, NaCl. And if you don't have enough salt in your diet, then you're going to have problems with this nerve impulse. Most Americans have the opposite problem. They have too much sodium. That's a whole other thing. So, when this neuron is resting, it has a negative charge. It is more negative on the inside than it is on the outside. And the reason for that is because of the work of something called the sodium-potassium pump, or the NAK pump. Now, what the sodium-potassium pump does is it pushes out sodium and lets in potassium. It pushes sodium outside of the cell and lets potassium inside of the cell. Now, both sodium and potassium have positive charges. And they have the same amount of a positive charge. They have one positive charge. But what happens is that for every sodium, or every potassium that gets let into the cell, a whole bunch of sodiums are shoved out. So eventually, there's not very many positives inside the cell, and that's why the charge is negative. Now, if you've ever played with magnets, you know that the positive end of a magnet and the negative end of a magnet they attract one another. Opposites attract in this case. And the same is true with these positive ion charges. What happens when all those sodiums get kicked out is they want to get back inside the cell because it's got a negative charge in there, and they're attracted to it. The negative charge, by the way, comes from chloride because it just kind of hangs out. It doesn't move around in this scenario. And so the sodiums want very much to get back into the cell. So instead of diffusing out when they are kicked out, they hang around the outside. That's the resting potential. It's negative inside because all the sodiums have been kicked out while a few potassiums have been let in, and so the sodiums are now hanging out, hopefully trying to get back in. Now, what causes this basic resting potential to get upset? Signals. Remember, dendrites collect signals. They either collect them from other neurons or they collect them from sense organs. And those signals essentially tell that receiving cell, that postsynaptic cell, to either let in more sodium or perhaps don't kick out quite as many or kick out even more. But in this case, it's let's not kick out quite so many. And so the interior of that cell body, that soma, starts to become more and more positive. Now, all neurons have what's called a threshold. In English, a threshold means you cross the threshold. It means that you're not here, now you're there. The threshold of a home means that you're now inside the home, for instance. Well, threshold here is kind of the point of no return. Once the charge of that neuron has reached threshold, whatever that threshold might be, once the charge has reached that threshold, then we're going to go from a resting potential to an action potential. Things are going to start happening. And basically what starts happening is very simple. Once that threshold is reached, there are channels in the wall of the cell, in the wall of the cell body there. These channels are called sodium channels because they allow sodium to pass freely back and forth through them. So what happens when that neuron reaches threshold, because those signals have come in, is those sodium channels open up. And all that sodium that has been waiting outside, desperate to try to get in, now starts to flood in. Okay, there is a lot of pressure that has been built up by all the sodiums pushing and shoving. And now once the door is open, they start to flood in. And so what happens to the charge in the inside of that part of the neuron? When all these positive charges come rushing in, it's going to become more positive. Now it not only gets to sort of a neutral, where there's just as many positive as negative, it actually gets even more positive than that. Because while the sodium's in the front, I'm anthropomorphizing like crazy here, okay? The sodiums in the front are saying, no, I don't want to go in, it's too positive. The ones in the back are pushing. Like, we want to get in. It's wonderful in there. We know it's wonderful. We want to. And so the charge doesn't just go from negative to neutral. It actually goes into the positive territory from all the pressure of those sodiums trying desperately to get in. Now, as I mentioned before, positive and positive don't get along. And the chloride's very happy that all these sodiums are rushing in, but what isn't happy is the potassium. Remember, we've got potassium in there from the sodium-potassium pump. And eventually it gets positive enough inside that neuron that the potassium leaves. It's too positive in there. The potassium is positive, the sodiums are positive, it's too positive in there. Like repels like, the potassium leaves. And once that neuron figures out that the potassium is leaving, it shuts those gates. It shuts them. Okay? It shuts the sodium gate, and it lets the sodium-potassium pump eventually return the neuron to its resting potential by kicking out a whole bunch of sodiums and letting a few potassiums back in. That's it. That's what happens. That is an action potential. We're at the resting potential. It's negative inside because the sodiums have been kicked out and only a few potassiums have been let in. We reach threshold because of signals from the dendrites. Sodium gates open. Sodium rushes in. The charge gets very positive inside the neuron. The potassium leaves when it gets too positive. The gates shut. The sodium gets kicked out. The potassium gets let in, and we're ready for another action potential. Now this happens incredibly quickly. We're talking dozens of times a second here. And it happens continually. It's why the vast majority of the energy used by your body is used by your nervous system. Okay? Because it's got to keep all of this going. And you're always having action potentials. Because your nervous system is a lot like a car. Well, actually, it's not a lot like a car. I take that back. Because a car you can turn off, right? This is kind of like a car that you can't turn off. It's always running. Now, sometimes it runs faster, sometimes it runs slower, but it's always running. And so you're always using energy. So yeah, you use a lot of energy to keep this going because this happens. I mean, think of all the neurons you have. Think of all the messages that get set dozens of times a second. There's an awful lot of stuff going on here, okay, in your nervous system. Now, the other thing I want to point out before I go into looking at the diagram is, in terms of how action potentials work, action potentials are always the same size. Either a neuron fires or it doesn't fire. You don't have little bitty action potentials and great big action potentials. The signals are sent by the number of action potentials you have in a certain period of time. Now, what we call that is all or nothing. Either you have enough of a stimulus coming in from the dendrites that you get an action potential, or you don't. Okay? So, now, how strong a signal is, for instance, coming in from a sense organ might be communicated by how many action potentials you have. So, you have a lot, it's a big signal. You have fewer, it's a smaller signal and such. But it's either enough to give you an action potential of a certain size, or you don't get an action potential at all. This is another way of looking at it. This is a diagram of what is going on during the action potentials and such. And what I will do then is go over, it's pretty much what I was describing, just more of a picture than words. Here we have resting potential. So there is no signal being sent. Notice that the sodium-potassium pump has already thrown out the sodiums, and it's a negative charge inside, positive charge outside. This will also, by the way, tell you what these nodes of Ranvier are for, these breaks in the myelin on the axon. So, signal comes in from the dendrites. Here we go. What happens is the sodium channels open up, including the ones here. You know, they open up on the soma. Sodium channels open up. Sodium comes rushing in. The interior of the neuron starts to become positive, doesn't it? Because these sodiums are rushing in the sodium channels. And what happens eventually when it gets too positive inside is the potassium leaves. And once the potassium leaves, then the sodium channels shut down, and the potassium then is brought back in. The sodium is thrown out, and we're back to resting. Now, this looks like it's happening through the myelin. This is not the best diagram, because it's not happening through the myelin. But it is happening, for instance, in these nodes here. Where it's insulated, you're not going to have any change. You're only going to have a change where the insulation is broken, right along here. So here we have a wave of positive ions coming down the pike. The positive ions hit this particular node of Ranvier, and they make the interior positive enough then for the sodium channels to open up. And the sodium comes rushing in. Sodium rushes in here from all over. It's rushing in. Is it going to want to go this way down the axon? No, that's positive now. That's where the wave of ions is coming from. Where does it want to go? Where it's negative. Where is it negative? Down here. So it starts to move down here. And eventually, because of this insulation, the wave of ions, the positive charge gets to here. What happens? The sodium gates open. Sodium rushes in. Does it want to go this way? No, of course. It wants to go that way. The myelin basically is the equivalent of throwing a baseball instead of running the baseball, or handing the baseball from person to person. You go a lot faster with throwing than you do with handing it off or running it. And so that but you can only throw a baseball so far so fast. The same way that baseball players will throw one to the other to make sure the ball gets to where it needs to be as quickly as possible. We have these nodes of Ranvier. So the continues, as we said, I hate to, at any rate. Sodium comes rushing in, eventually gets too positive. What happens? The potassium leaves. Look, the potassium is leaving. Potassium is leaving. It's blue. It's leaving. And so then what happens, of course, is the channels shut down. They bring the potassium back in, they throw the sodium back out, we go back to the resting potential. So you could have an action potential. You could have 3 or 4 action potentials going along this area here, going along this length of the axon at the same time. Because this area could be recovering from an action potential, the action potential could be here, and this could already be at resting potential waiting for the next one. So you could definitely, it's like having a rope and you're sending waves down the rope. Okay, you could have lots of waves on one particular rope, particularly if the waves are not very big. Lastly, here's yet another way of looking at an action potential. They're measuring the charge at one particular part of an axon. Okay? Resting potential, minus 70 millivolts. Here come signals from the dendrites. It's getting more and more positive. We reach threshold. The sodium channels open up. The sodium rushes in. The charge gets more and more positive. It goes past 0 because the ones in the rear are pushing and shoving. Right around here, the potassium leaves. The cell realizes that, shuts the sodium gates, and gets the sodium-potassium pump to start pumping out the sodium and letting in the potassium, pumping out the sodium and letting in the potassium. And in fact, it often gets so vigorous about throwing out the sodium, it actually goes lower than the resting potential. You ever been at a party where they were so busy throwing out the people who weren't invited that they wound up throwing out some people that were invited? It's kind of the same principle. And so eventually we wind up back at the resting potential. By the way, does this look familiar? It should. It looks like a heartbeat. Heartbeat monitors, essentially you have a nerve, I believe it's called the vagus nerve, V-A-G-U-S, that controls your heartbeat because all your muscles in your heart need to contract at once, or at least all of them in a certain area need to contract at once in order to pump blood effectively. Otherwise, you get what's called fibrillation, and you wind up with the muscles all working independently, and unfortunately that often winds up with the person not surviving. So, as I said, action potentials, very, very, very important.